Welcome back. We're doing the final block of protein purification, and we ended the last time with this slide here. We had gone through the three traditional liquid chromatography, size exclusion, ion exchange, and affinity. Affinity has a lot of variation, okay, because you saw what could be a ligand. Had a long list of things that could be. So the size of the columns, remember I think I said that the size exclusion may be 6 foot. Part of that is we need that distance for gravity. In other words, the pressure is just normal pressure. So right away, if you look at high pressure, high performance, then you realize that we're actually forcing a chromatography in a very short distance, i.e. speeds us up and it's more efficient. And so this right here is the chromatography column, and it has a metal casing, and there is a large amount of pressure from this pump applied. That pressure moves material through. This may be 8 inches, a foot long. It's a metal, and it was packed with various types of usually affinity resin. And so here's the solvent that we're going to pump through. And this is where we add our fractions. It's called the injector, and they mix with the solvent. And this is all punched through. And then there's a detector, catches the solvent, and the data, the actual protein or actual fragments of proteins are recorded. The amount here and the type, okay, the complexity. So in X and Y axis. So that's the principle. And so for the in silica folks, that means that every protein profile, that this is mass and charge usually, can be other things. Mass and charge, so it's M for mass, Z is for charge. Mass and charge. So now every protein can have a fingerprint, and that can be in the database. So eventually they can run all the proteins in your blood and figure out exactly what's there, okay, in a very short amount of time. So they can figure out exactly what bacteria, what virus you may have. Do you have cancer? I mean, that's the one stop fits all. It's just the effort, the time, getting this information is, we're not there yet. It's coming, but we're not there yet. So chromatography and reverse phase chromatography. So there's two types of this HPLC. One is called normal phase, and one is the reverse of normal phase. And of course, we like to give one thing extra names to trick you. And this is absorbance chromatography. It's also reverse phase. So this whole concept inside the column depends on the molecules of the proteins and what the matrix is on the column, because these matrix have specific ligands that have been chemically grafted or applied to them, kind of like they've been painted with these ligands that will catch some of these proteins. So you get the mobile phase solute onto an immobile N-alkyl hydrocarbon or aromatic ligand, and that's hydrophobic. So reverse phase uses and catches hydrophobic reactions. And so proteins that have a lot of R groups that are hydrophobic amino acids would be called reverse phase. Proteins that have low levels of hydrophobic, right? Hydrophobic would flow through quicker because they're not retained, whereas those that are retained tend to be hydrophobic. So once we kind of prep some of our protein fractions, we can now get a big separation on whether they're hydrophilic type proteins or hydrophobic. If a protein is in a membrane, it is by definition going to be a hydrophobic protein because it has to have at least 20 or more amino acids to span our lipid bilayers. So if you think about signaling, the receptors, these kind of things, they're going to be hydrophobic, whereas some of these other cytoplasmic will probably be hydrophilic. So in a signaling cascade, you're going to have a series of hydrophobic and hydrophilic proteins interacting. So here's the cartoon. This is a silica matrix here, which is sand or the glass. Here are these ligands, and it shows peptides that are caught, kind of like a mousetrap. These peptides are caught. Now you're saying, hold it, now we had proteins. Well, the proteins have to be chopped up because this technique requires smaller amino acid peptides, 3, 5, 6, 8, 10 amino acids. Hence, we get multiple fragments from every protein. So we get this fingerprint concept. Some can, depending on the size, we can bind the whole protein. So if they have a lot of hydrophobic character, certain proteins can be bound. Certainly peptides, nucleic acids can be separated. And it just depends on the peptide and the protein and the nature of the amino acids. Amino acids can have a lot of hydrophobic, or they don't have to have a lot of hydrophobic. Okay, so hydrophilic compounds are held and they're coming out last, eluted last. Hydrophobic are coming out first, and that's normal. Okay, normal phase. Reverse phase is hydrophilic compounds are eluted first. They come out first. Hydrophobic come out last. So if you take a sample, you can use either technique and you could separate hydrophilic and hydrophobic. And here's just a cartoon showing the mechanism. And so less polar, more hydrophobic analytes, that's the peptides, are more attracted, and more hydrophobic spend more time. So the more hydrophobic R groups, the more time you're spending on the column, versus the hydrophilic, you're not spending much time at all. And all the proteins are dissolved in methanol here. And so the time that a protein or a peptide is held on the column gives us good information about the nature. Is it hydrophobic or hydrophilic? And so we can get resolution of a sample and we can measure retention time. Resolution is a combination of separation between peaks from the columns. So here is the peaks from the columns here, right? And then the distance between the peaks is called the resolution, how far apart. And how thin are these curves? In other words, the tighter the curve, the more purification and the more pure it's going to be. And there are equations. We won't get into that, but you can do more equations here. You can add more polar solvent or water or less to manipulate proteins coming on and off the column. So there is some empirical here, empirical experiments to purify two similar proteins. So the length of the column, how you elute it, the pH of the column, and the ion pairing agents here are all variables that can be changed. So you can order different columns, different techniques for elution, pH, and whatnot. So a lot of empirical here. depending on the cell type, the organism. That's critical parameters. Okay. The last concept, and we're going to get back into this a little bit towards the end, but here's a cartoon showing separated proteins. They now have a machine that will take these proteins and apply them to this other machine right here, which applies trypsin and a buffer, and these proteins are chewed up. Okay, and then here's some more of the solvent, and it goes through these microreactors. So these proteins are being pushed through, they're being chewed into fragments, little peptides. At the end is a nozzle, and they're spit out as droplets, and each droplet has a peptide, and each peptide is detected by mass and charge. Each of these lines is a peptide here, and the amount of the peptide, this is high amount, this is low amount, and then more mass and charge, bigger proteins, smaller, bigger peptides, smaller peptides here. And so we have a timing that goes across here. So now every protein will have its own profile, protein fragment profile. So wild type proteins, cancer related proteins may have different profiles. They may be a little different. Maybe that's the key to the disease. But anyway, we can get a protein profile, and that's called electro absorption mass spec. So when you're measuring size and charge, that's said to be MS mass spec. And it's called electrospray ionization mass spec right here. And the protein is pumped in. There's a charge applied. It goes through a collisional activation here. So the droplets are separated, and each peptide is given a certain charge mass number, and this is size of the mass, and this is the amount. Okay, so you can get a profile here. And so you get a protein fingerprint using this mass spec. So now we can combine and actually get all this information all at once. Here's another cartoon showing that coming from the HPLC, it's neutral. They apply electrical charge, and these droplets have a positive charge on the ends, and the material tends to be in the middle, this little donut. And so what happened is it's spit out, ultrafine droplets spit out under this high voltage, and it vaporizes, okay? And so the ions can't go anywhere in the droplets. The ions are released, and they're detected by charge mass. And so now this is the mass spec you have. And so an ion having a mass spec ratio M versus Z, and the length of the bar is a relative abundance. So now not only can we do proteins, but we could do metabolites. How much GTP, how much ATP is produced. You know, in cancer cells, what's their metabolism? We can measure chemicals that are not even proteins, and it's called metabolomics. And so we can ask questions about the aspects of pathways here through mass spec. So it's called metabolomics, metabolism byproducts here, which don't have to be a metabolomics here. I can't spell, but we have metabolomics. Metabolomics is the process where we can study not only proteins and peptides and amino acids linked together, but also other kinds of chemicals in a living cell, and get an aspect of, say, glycolysis, Krebs cycle. We know about hydrogen ions and things that are produced under certain conditions. We can measure that. And here's just another one showing the different ones. And here's a protein showing not 3. And they had 25 different peptides that were generated from this one, it's pretty big protein, and they got about 28% coverage. So did I say the protein profile was perfect? No, but this is probably enough to identify this protein is not 3. And they're showing the different fragments in the different colors, and those fragments are shown in the mass spec ratio here. So just realize that, and this is another mass spec we'll talk about, Maldi-Toff, but bottom line is you're getting the mass charge ratio, and here's using different proteins, and you could look for these different fragments. So it's the 25 identified peptide cover 28% of the protein, and so the data suggests the protein that we think we're studying is the protein in the database. So last thing for bioinformatics, every protein theoretically can be in situ, virtually chopped up and made into peptides, and we can do a predicted charge mass profile. So even if nobody's ever done it, we can still have the predicted profile. If you did do it, this is what it is. And so now that is in the database. So it's experimental based, or if they've actually done it, then that would be functional based. So now this database can include both predicted charge mass ratio fingerprints or functional, where people have actually reported the data. So if you have a protein that's in cancer one way and maybe in non-cancer another, you can certainly study, what's the difference between the proteins, and look, is there, what are we missing here? Is there something going on in the two conditions? Just gives you a big insight here. So I think that's my 10-cent tour of protein fractionation. And so we're now going to move on later to the electrophoresis here. So I'm going to leave it with electrophoresis, and we will get the theory and how we do that. Okay, see you next time.